All right, so experiment 30 is isolation and identification of Streptococci. So Streptococcus bacteria are gram-positive, catalase negative cocci, and they're usually in chains. I don't really get why it says usually in chains in your lab manual, because strep means chain, and coccus means like circle, you know? So I don't really get why it says usually chains, but it says it nonetheless. So I'm going to repeat it in that manner. So yeah, several are pathogenic, so they cause scarlet fever, strep throat, meningitis, impetigo, whatever that is, and pneumonia. So humans have them, but humans do have them as normal flora in their mucous membranes, their mouth, their intestinal tract, and several are fastidious, which means that they need a lot of nutrients. Right? We went over that was part of like that one experiment, right? And they grow very well on blood agar plates. So blood agar plates, I think we're talking about that. Maybe, I don't know. Blood agar plates is like you use to like kind of mirror the, because they use like animal blood. So it's like they use animals, so it like mirrors your like cells in your body. So if it can infect an animal blood agar, then it can typically like infect, or if it can grow on a blood agar plate, it can infect like people, is the like theory there. But they use like a lot of nutrients. Okay, whatever. Okay, hemolysis. We do talk about it. So blood agar plates can show hemolysis. So yeah, we use blood to mirror the epithelial cells. So if you can kill, or if you can lyse the blood agar plate, then you can theoretically lyse the cells in the epithelial. So, but there are three different types of hemolysis, and that is gamma hemolysis, which just means no hemolysis, alpha hemolysis, which means that it's incomplete or partial hemolysis. I probably should have gotten a picture of this, but you can look that up, and there's pretty good examples in the lab. So, and the thing about alpha hemolysis is that this green color kind of appears. It's kind of green. It's like, it's green, but then it kind of, you have a red background because of the blood agar plate, and so it's like, it looks kind of grayish, but green, grayish kind of color. It's caused by the methemoglobin, which is the iron-containing molecule in hemoglobin. So anything that produces a green color like that is called a viridin, that word right there. Anything that causes that a green color like that is viridian. So if you have like viridian like bacteria, then you have alpha hemolysis. Last one is beta hemolysis, which is complete hemolysis. You'll see a complete clearing around the in the blood agar plate. And it's the major hemolysis of pathogenic streptococci, like the Streptococcus that cause scarlet fever, strep throat, meningitis, all those. Those are probably all beta hemolysis. So the way that they kind of infect, their virulence is, so they have substances that produce or enhance its ability to cause disease that are called virulence factors. All right, and bacteria use these virulence factors to facilitate growth. So killing human cells not only like helps like degrade the like immune system and stuff like that, and prevent that whole system from killing the cell, but then it also releases a whole bunch of nutrients that it can then absorb to grow. All right. So they use hemolysins to kill these bacteria, these human cells. All right, and so it's a type of virulence factor. Streptokinase is another type of virulence factor, and that dissolves fibrin clots. And fibrin clots can prevent bacteria from entering the body. So if you can dissolve these blood clots, then you could enter the body and like cause disease. But the good thing is that you can use these streptokinase enzymes clinically for people who have like blood clots in them if you need to like get rid of them. So that's kind of cool therapeutic use. Okay, so the growth of Streptococci. So most Streptococci are facultative anaerobes. So they grow well in air, but can grow without it. It's not necessarily needed. Some are microaerophilic. So they prefer high carbon dioxide concentrations and reduced oxygen concentrations, right? And then others are obligate anaerobes, so they only grow in the absence of oxygen. So a big thing to take away from this is that all of these microorganisms right here grow in anaerobic conditions, all right, or very low oxygen conditions at least. So we kind of took advantage. So what we did was we swabbed each other's mouth. That was always fun. And then we plated the swabs on a blood agar plate. We then sealed it with parafilm. And we put them all together in a candle jar, and we lit the candle, and what the candle does, it removes the oxygen and raises the carbon dioxide concentrations. So, and that promotes the Streptococci growth that we talked about last slide. And so what we observed is the type of hemolysis. So whether we had alpha, gamma, or beta hemolysis, we can kind of see that on the blood agar plate. Now, it's important to note that just because you have alpha hemolysis doesn't mean that you're sick or you have strep throat or anything like that. It's just kind of an interesting little fact you can spew around. But yeah, so that's pretty much the takeaway, is that we learned about the Streptococcus and how to isolate like that genus of microbes.